speaker in the session, Dr. Heinrich Haas, the Vice President of Drug Delivery of the BioNTech RNA Pharmaceuticals in Germany. He's going to talk us about the RNA nanomedicines for intravenous injection, control targeting selectivity to the organs. All yours.
So I'm still waiting. So I take the time to thank you for the nice introduction. I thank the organizers for giving me the opportunity to share our results with you. And I would like to thank the speakers before me to give some introduction into targeting and not targeting, which will be also a big part of the subject of this talk. I will be talking about RNA lipoplex formulations for intravenous injection for targeting selected organs, and this is all based on the RNA delivery platform technology we have developed have been developing since some years ago for new cancer immunotherapeutics, where we deliver messenger RNA to spleen and lymphoid organs for tumor immunotherapy. In this talk, I would like to give you a bit more details on the targeting coherences at the scientific level. Tomorrow, I will give a speech on the update of the clinical studies. So this is just a summary of the task formulation for RNA delivery has to fulfill. We have to deliver to the target, but the RNA must also be uptaken, translated, and so on. It has to be projected and all of this. This is just to place ourselves. We are not so much in the EPR and long circulating field. We don't work with specific ligands. We work basically, if you wish, with electrostatic interactions, which are also the foundation of many biological occurrences. Here, a slide which we showed already several times, where we demonstrate how we can direct targeting selectivity from one organ to the others by changing the properties of the lipoplexes we form from cationic liposomes and RNA. For example, in this case, by changing the ratio between the liposome and the RNA, and being more exactly the charge ratio of the cationic lipid and the RNA nucleotides. On this basis, we have selected formulations which specifically target the spleen, and this was the starting point for our clinical development for tumor immunotherapy. Today, I would like to focus more on the other end, where we could show as well that we target the spleen. And in the next slides, I will compare a little bit the formulations and the coherencies in spleen and lung targeting in a more detailed manner. So this is what we did. This was a study together with another group at the Mainz University, where Sebastian Rosichkeit, the PhD student, did all the work, and he was supervised by Ernesto Bockhamp. We more or less first reproduced an experiment which we showed you before, where we assembled positively and negatively charged lipoplexes by using excess of either liposomes or RNA, and we came up with positively charged lipoplexes, which we think look like that. Positively charged lipids at the outside, or negatively charged ones, where we think that the RNA at the outer membrane is responsible for the charge. We used DOTMAR as a cationic lipid and two different helper lipids, very simple, straightforward lipids, DOPE, and ethanolamine, which is widespread because it's phase properties, and cholesterol, which is also widespread. So very simple, straightforward systems, two charge ratios, two helper lipids, and beyond everything is the same. We assemble them and we inject them into mice. And this is what we get. So this is just for confirmation that the system works properly. So in each cases, we get transfection. This is luciferase coding RNA, which is injected intravenously, and then the mice are investigated by in vivo imaging. And you see we on each cases get signals, or the RNA is not degraded. If you take out the organs and look at them, we see that there is a certain profile or certain differences between the expression of the positively charged and the negatively charged ones, and the cholesterol and the DOPE containing ones. If we look at this into more detail, we can reveal more difference between the formulations. First of all, here we confirm for the negatively charged ones the preference for the spleen, and for the positively charged ones the preference for the lung. However, there are differences in the signal strength of luciferase of the extracted organs where the luciferase expression was normalized with respect to protein in the organ. So it's really a bit more a quantitative signal. Here, a question many people ask is how is with the delivery? Do you see also that the delivery of the vehicles is equivalent or not? And therefore, we have investigated the delivery of one vehicle component, namely the DOTMA, which is the cationic synthetic lipid, which is not occurring naturally. Therefore, you can easily extract it and determine it by liquid chromatography and mass spec LCMS. On the background of natural lipids. So we have quantified the DOTMA from the vehicle in the different organs, and what we found is with the spleen targeting formulations, we found a very big signal in the liver, but also in the spleen. So that's a spleen, but liver very big. Keep in mind that we didn't see any signal or almost any signal in the liver. So this is something which is contrary to other people who want to deliver material to the liver because, for example, if you want to do protein replacement and so on, this is the potential target of choice. For us, it's very important that we can reduce the signal in the liver and other organs as much as possible. On the other hand, positively charged ones, they go to the lung, and this is also where we got the signal. So first of all, very interesting, it appears that the physical delivery and the activity in vivo is not exactly correlated. Here you see this a little bit more to detail where we plotted the signal strength normalized per gram of organ for luciferase and DOTMA finder, and you see there's some correlation, but it's not identical, and therefore we have to understand this a bit more to detail. And here I would like to focus, as I said, exclusively now further on the positively charged ones where we got high amounts, similarly high amounts of DOTMA in the lung, but quite different signal strengths. What we did is we applied an assay to visualize the targeting of the RNA to the lung, which is our target organ, using a solvent-based tissue clearing method where you can really nicely visualize the lung, while if RNA has been delivered and translated, we get a blue color, and we can now identify to which parts of the lung the RNA has been delivered and expressed, and we see a certain quantitative as well as qualitative difference between the two formulations. So in the DOPE helper lipid case, everything is more confined to the central parts of the lung and not so much in the periphery, while with cholesterol it's more homogeneous and also the total signal strength is higher. This is also confirmed if you look more closely at macroscopic scale. There's not so much signal for the DOPE formulations, but much more for the cholesterol containing formulations, which gives us more clue to the molecular coherencies. So what we did then is to look more closely to the strongest targeting formulation, which is the COT.MA-COL formulation. And here you see which cells are targeted in the lung. First of all, we quantified the total amount of targeted cells, which is about 3%, and we could identify that about a bit more than 50% is endothelial cells, 40% blood cells, the rest is others. And now, of course, everybody would be interested in what do I get with the other formulations, and can I correlate now the targeting specificity of other formulations or the molecules in other formulations with the biological activity. Unfortunately, At this time, the PhD thesis came to an end, and so this is just, first of all, a feasibility study to demonstrate a method, but we think this can be very helpful for identifying and optimizing formulations. So summarizing up, what I showed was a combination of different methods to investigate the targeting specificity of RNA lipoplex formulations, where we found differences between physical and delivery and biological activity, where in particular we found information on the targeting selectivity on the cellular level. This is what we consider helpful for structure-function correlation studies, and this can provide as well an important basis for decision-making in pharmaceutical development. And with that, I would like to thank you for listening. I'm happy to take questions, and I would point your attention to the people who contributed. This is people from the preclinical development part in BioNTech. This is the RNA biochemistry group. This is our formulation group. And here the group of Sebastian Thosichkan and Nestor Bockham, with more people who contributed all to this work. Thank you very much.
Thank you very much. Unfortunately, we're running out of time. We have to close the session. I would like to thank all the speakers for, doing the effort to fit on the short time that we have for the presentations. Please, if you have any questions, I'm sure that they will be more than eager to answer you during the rest of the meeting. Thank you very much.